Let me start off with putting this particular lecture in context. So what we've done to this point in our discussion about molecular genetics, topics related to molecular genetics, is that we've described the molecular organization for the genetic material, the Watson-Crick model, understanding its structure in some detail. And so the importance of that, again, is to be able to relate to functions that are, uniquely associated with the genetic material. So the first function we've described is replication. So again, by understanding structure, you can understand how faithful replication of the information contained within the genetic material is attained. So now we're going to talk about a second function, and this is the idea of the genetic material being informational. The mechanism by which information is extracted from the DNA molecule, from the genetic material, and converted into some functional form. That functional form being proteins for the most part, not exclusively, but for the most part. So we're going to start to discuss gene expression. Gene expression is comprised of two steps or two stages, transcription and translation. We're going to be talking about transcription today. And so here are the readings for the next 2 or 3 lectures. Chapters 13 and 14 describe the process of gene expression, the 2 stages, transcription and translation. So again, as I said, the first step of gene expression is transcription. That's what we're going to focus on for this lecture. So this is simply the idea that transcription is the means by which, the first step by which the information is extracted from a nucleotide sequence within a DNA molecule, within a gene sequence. So again, the chain of command, as they describe it here, in terms of accessing and utilizing genetic information, is to go from DNA to RNA, another type of nucleic acid, and then ultimately to proteins, amino acid sequences, polypeptide structures. So transcription is that first step of gene expression where a DNA molecule or a DNA sequence, a gene sequence within a DNA molecule, is used to synthesize or create a molecule of RNA. Okay. So as we'll discuss, in terms of gene expression, protein production, ultimately what you find is that there's 3 primary types of transcripts that are produced in cells. There are hundreds of different types of transcripts, but the primary types, the major types, are what we call mRNA, rRNA, and tRNA. And they're all RNA molecules, but they all function differently within this cellular machinery that leads to protein production, that leads to the expression of genetic information. So that's what we're going to talk about today. The second step of gene expression, again, is the utilization of these transcripts to then produce a protein. So translation, the second step of gene expression, the actual synthesis of a polypeptide, a string of amino acids that occurs under the direction of mRNA and requires the involvement of the other 2 types of transcripts that I've described. The cellular machinery where this protein synthesis takes place, I've described previously, is what we call a ribosome. So we'll talk a little bit about ribosomes in the future as well. So here are those 3 primary types of transcripts produced off of gene sequences within a DNA molecule, within a chromosomal DNA molecule. And again, structurally, they're all RNA. So we've talked about, the nature, the biochemical nature of RNA, comprised of ribonucleotides, 4 different types, based on, the 4 different types of nitrogenous bases that you find in the nucleotides of an RNA molecule, A, G, C, and U, uracil, being unique to RNA, not present in DNA. So all 3 of these transcripts, again, are simply RNA molecules. They're strung together the individual ribonucleotides by phosphodiester linkages. There's a 5' end to the transcript. There's a 3' end to the transcript, and so on. And yet functionally, they're very different. As I said, each of these primary types of transcripts that you find in a living cell contribute different functions to protein production. So again, we'll talk a little bit more detail about each of these functions when we talk about the actual process of protein synthesis. But for now, what you can see here is the function of an mRNA is to carry DNA sequence information to the ribosomes. In other words, carry information that will tell the ribosome to create a particular string of amino acids during protein synthesis. So the tRNAs actually serve as the vehicles by which individual amino acids are carried to the ribosomes that can then be used for protein production. Okay, so again, remember there's 20 different amino acids that are used to construct proteins in living systems or living cells that that we're aware of and have characterized life as we know it on this planet. And then lastly, there's ribosomal RNAs as well. And these ribosomal RNAs are a component of the protein synthesis machinery in the cell, the ribosome itself. Okay, so this is just describing their general functions. We'll look at these functions associated with these 3 different transcripts in more detail when we talk about translation. So I do want to emphasize is that there are many other types of transcripts that can be produced in a cell. So again, RNA molecules produced off of a DNA template strand contained within chromosomal DNA. Other types, these less prominent types of transcripts in the cell, carry out more specialized functions, a lot of which is regulatory, determining how and when and at what level particular genes are being expressed in a cell at a given developmental state or stage. And then to give you another example, some of them are functional in other ways. And so just to give you an example, so we just talked about telomerase in a previous lecture. And again, what we said was that, there's an RNA molecule embedded within the telomerase enzyme that allows it to act as a reverse transcriptase. Okay, so where does that RNA come from? Well, it's a transcript. It's produced from a DNA sequence, a gene sequence contained within, chromosomal DNA of the organism. So there are many examples of this. As I said, hundreds of different types of these less prominent transcripts that serve more specialized roles. The 3 that we're going to focus on and spend some time discussing again are the predominant forms, the ribosomal RNA, the tRNA, and the messenger RNA, again, all of which contribute to the overall process of gene expression, protein production. So what exactly is transcription? How would you describe the process? We'll just run down this list real quick. The process of making an RNA copy of a single gene sequence, so making an RNA molecule off of a DNA template. So what are the genes? Genes are well-defined DNA sequences within a chromosomal-sized piece of DNA that give rise to these transcripts. So that must mean that they are subsets of base pair sequences within the overall DNA molecule that lead to the production of a transcript. And so what that means is that, again, in defining a gene, there must be a start site for transcription and there must be a stop site. So in other words, the entire molecule of chromosomal DNA is not transcribed, just subregions, individual genes. So genes have to be well defined at their start point, where transcription begins, and they have to be well defined at the back end of the gene where transcription is terminated. So we'll explore that a little bit as we go along. How are these transcripts produced off of gene sequences, off of these DNA sequences that represent individual genes? Well, there's an enzyme involved in the process. And we've already talked about the nature of that enzyme. It's an RNA polymerase. And more specifically, again, a DNA-dependent RNA polymerase. an enzyme that has the ability to read a DNA template to create an RNA molecule. So we've seen that. That's what the primase is in E. coli. And we talked about the role of the primase in DNA replication. So what does this RNA polymerase require in order to carry out its function? Again, it needs a single-stranded DNA template, and then it needs all 4 ribonucleoside triphosphates. So again, the building blocks for RNA synthesis are the ribonucleotides in the triphosphate form. And again, that's essential, that triphosphate form, in order to obtain the energy that allows for the formation of a phosphodiester linkage when you're polymerizing these individual ribonucleotides into these long chains that are the RNA molecule. So we've talked a little bit about the nature of the RNA polymerase. How does it read the template strand? Just like a DNA polymerase, it reads the template strand 3' to 5' so that it can create the new transcript in a 5' to 3' direction. So again, that has to do, the growth of the transcript of the RNA molecule always grows 5' to 3' because of the way that the enzyme forms that phosphodiester linkage. So that means, again, that the new bases that are incorporated into the growing RNA chain or RNA molecule are always added to that free 3' OH group. Okay, so again, that's something a concept that we've talked about previously. Okay, so getting back to the idea that genes, these transcriptional units within a chromosomal DNA molecule have to be well defined. In other words, the RNA polymerase has to know when and where to begin a transcriptional event. And it also has to know when and where to terminate a transcriptional event. So the two ends, the start point and the end point of a gene sequence, a transcriptional unit, have to be well defined so that the RNA polymerase knows where to start and where to stop. So there's actually a region of DNA called a promoter, which is the start site for transcription. So we'll talk a little bit more about the details of what these promoters look like as we go along here. So just to relate this concept of these promoter sequences or regions to things we've talked about previously. So, how do you initiate replication? It's at a specific site within the DNA molecule, the OREC region. And how does the helicase recognize that particular region of the chromosomal molecule of DNA? It's through consensus sequences. So it's the same concept here, is that within the promoter region, there are consensus sequences that are unique and uniquely recognizable by the RNA polymerase itself. So that's how the RNA polymerase knows the start point for a gene sequence or a transcriptional unit. Okay, so there's a couple of ideas I want to point out in this diagram here. So this is just a diagram, again, indicating a transcriptional event. So here's a double-stranded DNA molecule at the top. The two template strands held together by Watson-Crick base pairs. The two strands are anti-parallel to each other. All the common characteristics that we've described previously for double-stranded DNA. So the idea, moving down to this middle portion of this diagram now, is the idea is that on the left end here is where a promoter sequence is going to occur. That uniquely recognizable region that can attract the RNA polymerase enzyme to that site and initiate a transcriptional event. So the first thing I want you to think about is the idea is that, okay, so what happens is that when the RNA polymerase binds to that promoter region, the DNA has to be denatured. That's got to be part of initiation of transcription. But once that occurs, how does the RNA polymerase choose a specific template strand. There's two possible template strands. So what it's going to do is it's going to recognize the template strand that is oriented 3' to 5' relative to the catalytic center of the enzyme itself. So in this case, if the RNA polymerase is binding to the left here, it's the lower template strand that is oriented 3' to 5' relative to that catalytic center. So that's the template strand that is chosen as the template for transcription by the RNA polymerase. So what the RNA polymerase will do is, again, it'll read each individual base of each individual nucleotide in sequence one by one and incorporate a complementary nucleotide into the RNA chain that it's going to be producing. So again, if it reads a T, if it's reading this T here, it's going to incorporate an A containing nucleotide. T, it reads A, or reads T and incorporates A, reads A, incorporates U, reads C, incorporates G, and so on. Okay, so again, we've talked a little bit about that when we talked about the activity of the primase during a DNA replication event. So the end result is that, again, the RNA polymerase, once it begins to transcribe, it reads each nucleotide one by one within the template strand and incorporating the appropriate complementary nucleotide in the growing RNA chain. And what the enzyme does is it catalyzes formation of a phosphodiester linkage, 3' to 5' phosphodiester linkage, as we've described previously. So again, what that means is that the transcript that's being produced, following all of these parameters, is the RNA molecule is going to be growing in a 5' to a 3' direction. Okay, so here's a little bit closer view of what I just showed you. So now you can see things that are more molecular. So again, here's the double-stranded DNA molecule that represents the gene sequence. Here's the promoter to the left up here. The DNA becomes denatured ahead of the RNA polymerase that has been attracted to that promoter region. So now it has to discriminate between those two template strands. It recognizes the template strand, in this case, this lower strand here, that is oriented left to right, 3' to 5'. So as the RNA polymerase reads that template strand moving in that direction, 3' to 5', it begins synthesizing an RNA molecule in the 5' to 3' direction, incorporating a nucleotide one by one. So again, in this case, the very first nucleotide that's being incorporated into the RNA molecule contains G, because the complementary base in the template strand of DNA is C. And the second one that's been incorporated it reads T in the DNA template, so it incorporates adenine containing, a ribonucleotide with adenine. And then the next, it reads A in the template strand, so it incorporates U, and so on. So reading each nucleotide of the template strand of DNA one at a time and incorporating the appropriate complementary ribonucleotide in the growing RNA chain. So again, what you see is that the RNA molecule is growing in a 5' to a 3' direction. So the other thing I want to point out about this process of transcription is that what you notice is that the very first nucleotide that's incorporated into the transcript doesn't require bond formation. Okay, so what that means is, again, because you're using triphosphate forms of these ribonucleotides as the raw material for RNA construction, that very first ribonucleotide that's incorporated contains 3 phosphates at its 5' end. So that's a little bit unique. It makes the 5' end of the transcript chemically unique relative to the rest of the molecule. So just to move down to the other end here, so here's now the growing chain. You've incorporated G, phosphodiester linkage A, phosphodiester linkage U, phosphodiester linkage C. So now the RNA polymerase will read the T in the template strand of DNA, bring in ATP, right, ribonucleotide, 3 phosphates at the 5' end, and adenine at the 1' end of the ribose sugar, stabilized by base pairing between A and T, and then cleave off that diphosphate, as we've talked about previously, releasing energy that then leads to the formation of a phosphodiester linkage in that position. And that process continues indefinitely. Again, until you reach the other end of the gene, where there is information that tells the RNA polymerase to stop transcribing. So just like you have information, a promoter at the start point of transcription, you have information at the back end of the gene that terminates a transcriptional event. So the other thing I want to point out in this diagram is a little bit about terminology. So this template strand of DNA in the double-stranded DNA molecule that's used by the RNA polymerase, there's different names for it. Okay, so sometimes it's called the transcribed strand. Okay, well, that makes sense. It's called the template strand. That makes sense. That's what I've been calling it. And then it's also called an antisense strand or a non-coding strand. So those don't make sense, at least on the surface, or they shouldn't make sense to you on the surface. Because again, remember, what's happening during this process of transcription is you're extracting information contained within the nucleotide sequence of the DNA molecule and transferring that information to the RNA molecule. So antisense and non-coding suggests that the RNA transcript does not contain any information, when in reality it does. Again, an mRNA transcript, as I've described earlier, tells the ribosome what sequence of amino acids to generate, to create during protein formation. So the other strand of DNA within the double-stranded DNA molecule has names as well. It's the non-transcribed strand, the non-template strand. Okay, both of those make sense. But they're also called the sense strand and the coding strand. So in other words, the information is contained in the non-template strand of DNA within the gene sequence. That doesn't seem right. Okay, so where do these names derive? Well, simply the idea is that if you look at this, remember the template strand for RNA polymerase is running left to right, 3' to 5'. And the RNA transcript that's being produced is being produced, left to right in a 5' to a 3' direction. So that's the exact same direction as the non-template strand. Remember, this strand up top here that is not being transcribed is oriented 5' to 3' left to right, just like the transcript itself. And both the transcript as well as that non-template strand have the ability to stably base pair with the template strand used by the RNA polymerase, the template strand of DNA used by the RNA polymerase. So in other words, the transcript itself and the non-template strand in terms of nucleotide sequence are exactly the same, except for the fact that in RNA you have uracil containing nucleotides, and in DNA you have thymine containing nucleotides. But both of them, in terms of their nucleotide sequence and informational content, are essentially the same, except in RNA, again, you have U's, and in DNA you have T's. So that's where these names derive. Okay, so let's talk a little bit about the nature of the RNA polymerase itself. So again, RNA polymerase was first purified and characterized from E. coli, just like the DNA polymerases that we've discussed previously. And what did they learn about the structural characteristics of the RNA polymerase? Well, it's a complex, just like the DNA polymerase 3 that I've discussed previously. In this case, there's 4 different subunits. There's 2 alpha subunits. There's a beta subunit, different polypeptides. There's a beta prime, and then there's a sigma subunit. So all of these things aggregate into a complex that then functions as an RNA polymerase. So what do? Well, the alpha subunits seem to be primarily structural. They provide sort of a framework upon which all of the other subunits can hang, and then allow them to carry out their specific functions. So the beta subunit is the subunit within the complex that has the RNA polymerase activity, the ability to catalyze the formation of a 3' to 5' phosphodiester linkage. Okay. So what does the beta prime subunit do? Well, the beta prime subunit confers general DNA binding activity to the complex. So for an RNA polymerase to work, it has to find a gene sequence. And again, the start of a gene is going to be a promoter region. But how does it find chromosomal DNA just in a general form, or in a general way, so that it can bind to it? Well, that's through the general DNA binding activity of that beta prime subunit. So once it binds to DNA, how does it find the promoter? Well, that's what the sigma subunit allows for. The sigma subunit has specific DNA binding activity. It has the ability to recognize consensus sequences within the promoter specifically. So the RNA polymerase can then associate with that promoter region and initiate a transcriptional event. So here's, again, a little bit of a written narrative describing the process of transcription. You can read through this yourself. We'll just go through it quickly here. So when you describe this process, just like I described DNA replication, is that you break it up into different stages. So initiation, elongation, and termination. Okay, so initiation, again, requires the recognition of the promoter within the gene sequence by the RNA polymerase. So once that binding occurs, then transcription can begin. The gene can become transcriptionally active. Okay, so as part of that process, there's a requirement that you have to unwind and denature the DNA just ahead of the RNA polymerase so it can find the template strand of DNA and begin the process of transcription, begin the process of RNA synthesis. So once all of those events occur, as part of initiation, and you get the process started, then you have, the bulk of the work being done during the elongation stage, when the RNA polymerase is just reading down the template strand, extracting the information through the creation of the RNA transcript. And then lastly, you have to terminate the process or it'll continue indefinitely. So how do you stop it? So again, at the other end of the gene, the back end of the gene, you have consensus sequences that serve as terminator sequences. And they dissociate the RNA polymerase from the template strand of DNA to terminate the transcriptional event. So again, just to relate RNA polymerase to the characteristics that I described for some DNA polymerases is the concept of processivity. So RNA polymerase has very high processivity. Once it's bound to DNA and it begins a transcriptional event, it can hang on to that template strand for long periods of time. So high processivity. And it's hard to get it to release. That's why you need those terminator sequences. Okay, so again, this is just a diagram that here's initiation, here's the RNA polymerase, there's the promoter region, binding, transcription the DNA is denatured, a single-stranded template is presented to the RNA polymerase complex, transcription then proceeds, and then now you've transitioned from initiation to elongation. So then the bulk of the work is being done, the gene is being transcribed, an RNA transcript is being produced, the free end is the 5' end, the growing end is the 3' end, and eventually the RNA polymerase at the other end of the gene will run into these terminator sequences and the process will stop. So let's look a little bit more closely at the promoters. So what is it within the promoters? What sequences, what consensus sequences are uniquely recognizable by the RNA polymerase? There's actually two of them, and they're described here. So these are what are generally referred to as a -35 and a -10 region within the promoter. And they're specific consensus nucleotide sequences. So here's another representation of the same thing. So what we have in this diagram here are the alignment of the upstream region of genes from the E. coli genome. And actually what you find is in this list is that there are all three types of genes. So what do I mean by that? Three different types of genes that produce the three different types of transcripts that I described earlier. So protein encoding genes produce mRNA molecules through transcription. tRNA genes produce tRNA transcripts, and ribosomal RNA genes produce rRNA transcripts. So all three of those gene types are aligned in this diagram here. So many of these are protein encoding. You can see here's a tRNA gene, and these three here are ribosomal RNA genes. And what they've done is they've aligned them. They're the nucleotide sequence of, actually this is the non-template strand. So the green box indicates the transcription start site, the very first nucleotide that's read by the RNA polymerase during a transcriptional event. Okay, so they're all aligned, and when you align them and you go upstream, in other words, more towards this is the 5' end of the DNA sequences to the left. Remember, I said this is the non-template strand of the DNA of the DNA sequences, of the gene sequences. This is the 5' end to the left, the 3' end to the right. So when you do this sort of alignment, what you can see is that there's a region here that's been highlighted in color that serves or represents one of those consensus sequences. This is the -10 region, or more formally referred to as a Pribnow box. And you see it's the same for all of these different genes. So that's a sequence within the promoter that is uniquely recognizable by the RNA polymerase. And again, remember that recognition is conferred through the sigma subunit. So there's another consensus sequence within the promoter. This is what we call the -35 region. So again, it's in this colored region up here. And you can see when you compare all of these different genes for the Pribnow box and the -35 region in terms of a nucleotide sequence, they're not identical, but they're similar. So again, that's true for all consensus sequences. They're similar, but they're not necessarily identical. But the general consensus for the Pribnow box for most genes within the E. coli genome, the sequence is going to be TAT-AAT. And for the -35 region, TCT-TG-ACAT. Those are the consensus sequences. And again, both of them are uniquely recognizable by the sigma subunit of the RNA polymerase that allows for initiation of a transcriptional event off of each of these genes at this green boxed nucleotide. So how does initiation occur? So here's your RNA polymerase with the associated sigma subunit here. Remember, that's the one that confers recognition of those consensus sequences. So the complex can bind to double-stranded DNA in a general way using the beta prime subunit. So then once it binds to double-stranded DNA, it can move along that structure until it finds a promoter, and the sigma subunit recognizes the promoter. So what happens is that there is a clamping down or a tighter binding that occurs through that recognition process. So what happens is that through the sigma subunit, the RNA polymerase complex will clamp down more tightly on the DNA molecule within the promoter and twist and distort it similar to the helicases that I described for initiation of DNA replication. So when that happens, when you twist and distort the double-stranded molecule of DNA, you open up a short region of denatured DNA, two single-stranded templates. So a transcriptional bubble. Okay, simply by twisting and distorting the DNA. So what allows for that? Actually, if you go back to the Pribnow box, you see it's rich in A and T. And remember, A and T base pairs in double-stranded DNA are only held together by 2 hydrogen bonds. So it's easier to disrupt than a stretch of double-stranded DNA where you have lots of GC base pairs. So by binding to this promoter region, the RNA polymerase complex, you twist and distort the DNA such that it now opens up from the Pribnow box and up through this region that has that transcription start site, that +1 nucleotide. So that's what they're trying to represent here. So here's the promoter to the left of the diagram. The RNA polymerase is clamped down, twisted and distorted the DNA, opened up a transcriptional bubble, and one of the 2 template strands has been chosen for use by the RNA polymerase. So it must mean that from left to right, it's oriented 3' to 5'. So then the RNA polymerase will read each nucleotide one at a time and incorporate a ribonucleotide into the growing RNA chain that it's producing. And again, by producing those phosphodiester linkages. So that's initiation. So what happens is that after you create an RNA molecule off of that template strand that's about 8 or 9 nucleotides in total length, you no longer need that sigma subunit. The sigma subunit only is important for initiating a transcriptional event. So actually what happens is that it dissociates from the RNA polymerase complex. It dissociates and becomes free. What you have is what we call a core enzyme, the core RNA polymerase, that then does the bulk of the work during the elongation phase of transcription. So again, the gene, once that sigma subunit dissociates, the RNA polymerase with that characteristic of high processivity can begin to transcribe through the bulk of the gene sequence and create that RNA transcript. So how do you stop it? So how do you terminate? So again, there must be signals, must be consensus sequences at that 3' end of the gene. So there's actually, in E. coli, there have been 2 mechanisms that are used for transcription termination. And they're very similar to each other, just subtle differences. And I just want you to understand them in, a general way. So this is, one of them is protein-dependent termination. One of them is protein-independent. And the name of the protein is what we call the Rho protein. So let's look at these 2 mechanisms. This is Rho-independent termination. So without the involvement of a protein. So here's the RNA polymerase, the core enzyme bound to the template strand, creating a transcript. Again, to the left is the 5' end, to the right is the 3' end. So what that RNA polymerase has done at the back end of the gene is it has transcribed through terminator sequences. And those terminator sequences, represented here as a yellow box and a green box, are transcribed. They become part of the transcript itself. So how do they lead to termination? Well, the idea is that they exhibit base pair complementarity to each other. So they're GC-rich regions or GC-rich nucleotide sequences that exhibit base pair complementarity to each other. So they base pair with each other. And so what that does is it leads to the formation of what we call a hairpin loop in the transcript. So secondary structure. The rest of the RNA molecule is linear. It's just a linear RNA molecule. But in this case, where those terminator sequences are present, you get the formation of this secondary structure, this hairpin loop. And the formation of that hairpin loop is disruptive. In other words, it starts to weaken the hold of the RNA polymerase to the template strand, decrease that characteristic of processivity. And so eventually what happens is that the RNA polymerase falls off. And no longer being associated with the template strand, it stops transcription. So row-dependent termination is essentially the same, except in other words, it involves the formation of secondary structure within the transcript itself, but it also involves a protein, and more specifically the row protein. So here's the RNA polymerase transcribing through the gene. We're at the back end of the gene now. You transcribe through a region. In this case, instead of forming a hairpin, what's called a lollipop structure, part double-stranded, part single-stranded. But that secondary structure is disruptive. It weakens the hold of the RNA polymerase on the template strand DNA. So in addition to that, there is a row protein that has the ability to bind. Its characteristic is that it can bind to single-stranded RNA molecules. And in doing so, they associate, the row protein, subunits of it, will associate with this complex of RNA polymerase and newly formed transcript in the area where that secondary structure has formed. And all of those interactions, the row protein with the secondary structure, weaken the hold of the RNA polymerase on the template strand, and so it releases. And once it releases the template strand, transcription is terminated. So you can see the two mechanisms are very similar, just one of them involving the row protein and one of them not. Okay, so again, just to emphasize is that these transcriptional events that I've just described, as initiation, events unique to initiation, elongation, and termination, they're all in E. coli. These transcriptional events are the same for the 3 different gene types, major gene types, that you find within the E. coli genome. So again, what that means is that protein encoding genes are transcribed by RNA polymerase, the E. coli RNA polymerase, the same as ribosomal RNA genes are, and the same as tRNA genes are. That's why the promoters are the same for those 3 different gene types within the E. coli genome. Okay, so I'm going to stop there and we'll pick up next time to further this discussion on transcription. And there we'll look at, again, some similarities and differences with the way that transcription proceeds in a eukaryotic system.